Hey everyone, welcome back to DIY Biotech. My name is Nick. I'll have my PhD in less than a week in bioengineering, where I did genetic engineering on yeast. And today I have a special treat, as always. This is completing the trifecta. We looked at psilocybin production in yeast, we looked at cannabinoids in yeast, and now we're looking at producing opioids in yeast. And this is all just an elaborate trick to make you guys learn about metabolic engineering, which is something I'm really passionate about and really interested in. And I'm tricking you into my proverbial van with drugs. So let's go along for the ride. This paper is called Complete Biosynthesis of Opioids in Yeast. And the sort of PI, the principal investigator of this paper, now the PIs are listed last in papers, is this lady, Christina Smolk. She is really cool. She's a professor at Stanford. So I'm going to just show you a little bit of background on her. So she started this company called Anthea maybe 5 years ago, maybe 10 years ago. I don't think they're very clear about exactly what product they're making, but I think we're all pretty sure that it's some sort of opioid that they're making in yeast. There's a master's student that was in my research lab that now works at this company. And of course, he signed an NDA, so he hasn't told me what they're producing. How's it going, Chris? She's also done some other stuff with producing opioids in yeast and a lot of other metabolic engineering work. Christina Smolk is probably up there with Jay Kiesling, who did the cannabinoid in yeast paper in the last video. But she has worked with this now PI, Yan-Ren Lee, who is at UC San Diego now, I believe. But she was at my university, and she worked her lab was across the hall from mine. So she was on my advancement to candidacy committee. And so that's sort of my relation to Christina Smolk, sort of secondhand relation. So that's sort of the background, the lore around this paper, who's in this paper, but sort of the background of producing opioids in microorganisms. Around the same time that this paper was published in the journal Science, this paper was published in the journal Nature Communications, which is maybe one rung below Science. And this is producing opioids in E. coli. So we aren't going over this paper because I'm a yeast supremacist. So we're going to look at this yeast opioid paper instead. And this was published about the same time as the E. coli paper in the fall of 2015, again in the journal Science. So it's got to be a good journal if the entire journal title is just Science, right? So Science is right up there with Nature. Very big, broad journal for all different kinds of research. So anyway, again, we're making opioids in yeast, and so we'll go through the paper. This is a very, very dense metabolic engineering paper. Again, just like the previous two articles that I reviewed, it's pretty traditional metabolic engineering in a few ways that I'll highlight. So we'll go over the abstract first very briefly, and then over the figures, and then some of the details in the paper. And just go over some techniques that they used here in this paper. So first, what they set this up as yes, obviously we can make opioids in poppy plants. However, it's a crop, it's a natural product. And so there's variation year to year. Weather can change, how much you produce. And that makes things expensive. You have to use land resources, you got to use water resources. Setting up the argument for let's make it in yeast instead. So we engineered yeast to produce selected opioid compounds, thebaine and hydrocodone, starting from sugar. So again, the term here is de novo biosynthesis. So from nothing, we're making these opioids, so just feeding sugar. Whereas lots of other previous work have done conversions on opioid precursors, but they haven't produced opioids in yeast from the crude sugar. This pathway is the most complex pathway that we've looked at thus far. The psilocybin pathway was really big and long, and then the cannabinoid pathway was really difficult because it was coming from plants. And then this pathway is also coming from plants, mostly, but they're looking at expressing 21 to 23 enzymes depending on what opioid they're producing. And just for some context, when I'm doing genetic engineering, when I want to put a gene in a yeast, it takes about a week, best case scenario. More realistically, it takes something like 2 weeks to do that. They're saying they expressed 21 to 23 enzymes, all from plants, mammals, bacteria, and yeast. So from all of these different kingdoms of life, they're expressing in the yeast. And so there's a lot of challenges there, and we'll go over that. So if you're starting from zero and you want to try to make opioids or any compound in yeast, you search your compound on Google and put KEGG after it, K-E-G-G. And you find the pathway for the molecule. So I'll show you that just very briefly. But I put in thebaine, I think that's how you say it, KEGG. And then here's the compound listing. Sure enough, there's our compound, there's thebaine. We can go to the pathway, one of the pathways for producing thebaine right here. And you can see from L-tyrosine, so just from this amino acid tyrosine, we do lots and lots of conversions, lots of enzymatic steps. Keep going. There's thebaine, right? So lots of enzymes to get there. Down on the bottom here, there's a familiar opioid, morphine, and another familiar one, codeine. And you might be looking at this pathway and recognize some other compounds as well. So this is isoquinolone alkaloid biosynthesis. So that's the pathway here that we're looking at. So these isoquinolone alkaloids. So let's continue. That's the abstract. In the PDF version, they put the figures after the references for some reason. I'm not really sure. But as you can see, this paper is very dense with enzyme engineering and genetic engineering for putting these enzymes in yeast. So moving enzymes from another organism to yeast is not as simple as copying and pasting, right? There's a lot of pieces of enzymes that may be specific to plants, for example, that don't function in yeast. And we'll look at some of those details as well. All right, so going through the figures first, because I think that's the easiest place to start. Usually when I break down a paper, or I think in general when you read a paper, especially if it's very new to you, reading the abstract is a really good idea, reading the conclusion is a really good idea, and going through the figures is a really good idea, really in whatever order you want for those three things. And then once you have the big picture idea, you can go through the details and the results and conclusions of the paper. And then if you really are going to actually recreate the data in the paper, or you want to steal some of the methods, then you go to the methods section and read that. All right, so here's figure one, and it's showing just a different version of the KEGG pathway that I already showed, and it's showing from sugar, you go through this shikimate pathway. I believe it's the shikimate pathway. Yeah, the shikimate pathway to get to tyrosine. So they're saying that they had to overexpress or modify these enzymes in this pathway to produce more tyrosine. And again, tyrosine is just an amino acid, and it's also a precursor to a lot of other biochemicals. So there's been a lot of metabolic engineering work on how we can make more tyrosine in yeast and bacteria and whatever organism. And so they looked at their previous research and research from other groups to make more tyrosine. So then from tyrosine, you get to L-dopa and then to dopamine. Yes, that dopamine. Dopamine is a precursor to opioids, which is just interesting to think about. And then all of these green steps are cytochrome P450 enzymes. So bacteria don't naturally or can't naturally use P450 enzymes. They require some extra cofactors that are only present in eukaryotes. And a lot of these P450 enzymes are made to be compatible only in like plants, for example. And so you need extra machinery in order to use those P450 enzymes. So anytime you see a green arrow, think that there is additional complexity in that step. There's all of these enzymes that they have to add, a lot of cytochrome P450 enzymes from plants that they're adding, going through this pathway. Finally, they get to thebaine, which is a precursor to other opioids. And then finally, hydrocodone is the last product that they go for. So again, 21 to 23 enzymes in order to get just very small amounts, and we'll talk about how much is small, but very small amounts of these opioids. All right, so let's go into the paper, and then maybe we'll look at the other figures as well. Traditional metabolic engineering paper, they're saying chemical synthesis of these opioids is not commercially competitive. So you can't take some petrochemical and convert it to opioids efficiently enough to be competitive with just growing the poppy plant, which is wild to think about, but that's the case. The class of compounds that we're working with here are these benzyl isoquinoline alkaloids specifically, or BIAs. So if you see that BIA, it's just the class of compounds that we're working with in this paper. And then of course, there's so many enzymatic steps. They're having this little caveat that every step that you add in this pathway is going to decrease titers. So like if your first enzyme is 10% efficient and then your second enzyme is 10% efficient, then that third compound that you get is only going to be 1% efficient from that starting material. And so having, highly efficient enzymes is really important. And what it also means is that the more steps you add, the lower your yields are going to be at the end. Another really interesting thing that wasn't present in the psilocybin paper or in the cannabinoid paper is they have a lot of talk about getting permission from the DEA, and that there were very strict regulations in them producing these compounds, and even regulation around the strains that they made. So a lot of people in the comment section are saying like, what if we had this yeast? Could we grow it at home? Whatever. This is the first paper we've looked at where they actually address those concerns. So they say that they got special permission from Stanford to do this. They, worked with the DEA, the U.S. Drug Enforcement Agency, to work on this project. All of the researchers had background checks. There were detailed protocols on how you can use the yeast, containment of the extra containment of the special strains, laboratory security, and management and reporting protocols. So they were really serious about locking down these strains. And they have more caveats at the end. But basically, these strains are essentially useless because they produce so little opioids, but they still had all of this regulation around it. So then they're ready to start putting genes into their yeast. And you can see that here they were integrating their genes in chromosomal regions from which we expected no growth defect and active expression as integration loci. So, in a given genome of an organism, you have regions that are actually functional, but you also have regions that don't do anything. It's sort of nonsense just sitting there. And then you have regions that are sort of closed off and not functional. Even if you tried to put a gene there, they wouldn't be functional. So they're saying that they want to choose a region of the genome that is open and ready for genes to be transcribed, but that region also doesn't have existing functional genes. And we're working with Saccharomyces cerevisiae here, which is baker's yeast or brewer's yeast. So we know a lot about this yeast. Of course, the genome is completely sequenced, and we know a lot of examples of regions in the genome where we can integrate these genes. So that is an important step. Then they say that they want to increase the accumulation of L-tyrosine and this 4-HPAA molecule, because these are precursors to these BIA compounds. And we looked at that in figure 1. They want to increase flux to reticulin. So reticulin is one of the intermediates of producing these opioids. And again, flux is that movement through a particular pathway. So we want to push, mass or moles or numbers of compounds into the opioids as much as we can, while restricting flux to side pathways, right? And again, we're going through this article not in the specific details of what genes they used, because you can see those genes in KEGG or read through the paper if you really care. What I think is the interesting points here is the techniques that they used and sort of the background knowledge that you have to have in order to understand what's going on here. So one of the another technique that they use and another concept here is this LCMS method, which LCMS is liquid chromatography with mass spectrometry. It was used in the psilocybin paper. It was used in the cannabinoid paper. It's sort of the gold standard chromatography for molecules that you're only getting very small amounts of. It's able to detect down to microgram per liter range of compounds, whereas just liquid chromatography without the mass spec can only detect around like a milligram per liter usually. So the liquid chromatography with mass spec is just the liquid chromatography is basically you're passing your sample through what we call a column, which is just a tube filled with a particular material that can sort the compounds that are going through. So certain compounds will stick to that column better than other compounds. And so on the other side, you have individual compounds, hopefully individual compounds coming out one at a time. And so you have a detector on the other side that sees those compounds coming off and generates these graphs with peaks. And if your method is the same every time you run the liquid chromatography, you're going to get your compound of interest coming out of the column at the same time every time. And so with LC-MS, your detector is a mass spec, and that's a whole other topic, but basically you ionize particles, you ionize your compounds coming out of your sample and shoot them through a really strong magnetic field, and that allows sorting of those broken up compounds by their mass and by their charge. And so universally, doesn't matter where you are in the world, doesn't matter what mass spec you're using, if you send like a caffeine molecule through a mass spec, it's going to create a very distinctive readout. And so even if you're producing a compound that you don't know what it is, the mass spec can tell you what that compound is based on how it breaks up in the ionization and how it's separated by the strong magnetic field, which sorts based on size and charge. Anyway, that's a tangent. LC-MS is really cool. Chromatography in general is super, super cool. But that's the method they're using here to detect their really tiny amounts of opioids that they're producing. And their titers in the beginning for producing reticulin were 12.3 micrograms per liter. So they say later that opioids are usually given in doses in the milligram range. So you think about, again, a liter volume. This is upstream, like 8 enzymatic steps away from the opioid that we want to produce, and they're only getting 12.3 micrograms per liter. So you would need to chug 100 liters of this culture in order to get close to a dose of reticulin. And reticulin isn't even the drug that you're going after. It's 8 or so enzymatic steps away from reticulin. So in this paper, we're not going to get industrially relevant titers, and that's talked about later as well. Okay, again, they're bringing up these cytochrome P450 enzymes, and that's a big difficulty. They use this tool called BLAST, and I really want to show this to you guys. This is super cool. So look, if we want to see this enzyme that converts codeine to morphine, we can click on it, and then we should be able to see the sequence as well. So we're going to go to, okay, so here's a gene from opium poppy, and here's the nucleotide sequence, and here's the amino acid sequence of that enzyme, right? So potentially this enzyme doesn't work very well in yeast, and so we can go to BLAST, NCBI BLAST. We can look up this nucleotide sequence, or let's actually look up the amino acid sequence. Okay, so we're on BLAST, and we have the amino acid sequence of this codeine to morphine enzyme from the opium poppy, and we can actually look up this protein sequence and see if there's similar enzymes out there. So I'm literally just pasting the amino acid sequence in here and searching it. No login required. So you would use this in a case where the plant enzyme doesn't work well in yeast, and so you want to find an equivalent enzyme that might still do that enzymatic step from some other pathway. And there's a good chance we don't find anything of interest here, but I just wanted to show you guys what you can do with NCBI BLAST. All right, here we are. We got our search results in about 30 seconds. And so this right here is from the opium poppy plant. This has 100% similarity. So this E value, the lower it is, the more similar it is. So a lot of these enzymes are from the opium poppy, and potentially they're similar but not the same enzymes. So you could try out different enzymes from the same plant, and maybe one works better than in yeast than another. So these are all from the opium poppy. This is from another organism. Let's see what this is from. This Vitis riparia. Is that grape or something? Let's see. Yeah, so riverbank grape. Right, this is insane. So if you're doing this metabolic engineering and you find an enzymatic step that isn't working well for you, then you can copy that amino acid sequence from these publicly available databases, put it into the BLAST search system, and find similar enzymes that probably have that same activity. This is sort of the easier step relatively. Maybe it requires a little bit more background research. Actually implementing these genes does take a long time, and comparing lots of genes does take a long time. But it's crazy that you can go in and search for these enzymes. So apparently this grape has an enzyme that converts or may convert codeine to morphine, or it has similar function to an enzyme that converts codeine to morphine, which is really, really, really crazy to think about. So anyway, they use this as a tool here a lot in this paper, I'm sure, in order to find enzymes that may work instead of enzymes actually from the poppy plant. Another thing that they had to do is what's called codon optimization. I believe I have a video of that on my channel, but the idea with codon optimization is that, we have codons in our DNA sequences. So there are 3 base pairs of DNA is a single codon that codes for a particular amino acid. So this is like the language of life, right? So for every organism, just about the same 3 codons codes for the same amino acid. However, every organism speaks a slightly different dialect of this language of life. Especially when you're moving from different kingdoms of life. If you're moving from one yeast to another yeast, you probably don't have to do codon optimization, but it's recommended, right? So, yeast speaks, one particular dialect of the language of life, and this opium poppy speaks a slightly different dialect. And so there are tools called codon optimization tools that perform that translation for you, right? So of course they had to use this a lot in this paper as well. Okay, we're gonna skip over this part. I don't like that part. Another tool that they used here is Western blot analysis. So you can take your gene of interest and put it in your yeast. Just hope that that chemical conversion that you're expecting does happen, and hope that you get that chemical. But often it doesn't work, right? So how do you make sure that you're even making that protein properly in the yeast? And you do that with Western blot. So if you're familiar with electrophoresis gels for DNA, so like PCR and then running an electrophoresis gel, a Western blot is basically a way of separating different sized proteins and different charged proteins in a gel medium. So it literally feels like a gel, like Jello or something like that. So you run a really high voltage through this gel, and on one end you put your protein, and based on the size and the charge of that protein, it's going to move at a different rate through the gel. So you get separation of your proteins, and you can say, I expect my opioid biosynthesis protein to be 100 kilodaltons in size, and at that 100 kilodalton range where I expect that protein to be in the gel, sure enough, it's there. And so they use that a lot here to validate that they're actually making the proteins properly. And they actually did have a lot of problems there, because they're moving from plants to yeast, right? There's going to be misprocessing of these proteins, so misfolding of these proteins, or there's downstream protein modifications that happen that may not be happening correctly, since you're moving from one kingdom of life to another. And that was a big problem that they faced with a few of the enzymes in this paper. Finally, we get to the conclusion. That was sort of the meat of the paper. Again, if you care about the individual enzymes, you can look at the individual enzymes in the paper, but I think the sort of understanding the tools that are used here is the most important part. Also, this last bit, they were able to produce 0.3 micrograms per liter of hydrocodone. So again, very low levels. And then here they were producing about 6 micrograms per liter of thebaine. We're not having this anytime soon. That being said, this was about 8 years ago, so maybe the Anthea company is producing a lot more of these opioids now. So they say this work represents the complete biosynthesis of opioids in heterologous host starting from central metabolism. So again, this is de novo production of these compounds, and heterologous meaning from another organism. So yeast don't naturally make opioids, poppy does, and so we took enzymes from poppy as well as other organisms and made it in yeast. So that's heterologous. Again, they're flexing that they expressed 21 heterologous enzymes from plants, mammals, bacteria, and yeast. Overexpression of 2 native enzymes, so 2 enzymes that were already present in yeast, they overexpressed, and they had to delete 1 yeast gene. So lots of modifications in this single strain. Typically, when we're doing metabolic engineering, I would say something like 5 to 10 modifications is normal to produce a compound. But looking at, dozens of modifications is a lot of work. There's a lot of work that goes into this. This paragraph is really cool and really interesting, and I wish more papers would talk about the practicality of the work that they're doing, be a little bit more realistic. So they say here, over a 100,000-fold improvement in fermentation titer to get to about 5 grams per liter would be required for yeast-based production of opioids to be a feasible alternative to poppy farming for commercial production. This is fascinating. They're actually saying like the limitations of their work in this paper. 100,000-fold means they need to make 100,000 times more product than they're currently making, which isn't impossible, right? This gram per liter range, we've done it for other compounds, but not for opioids, right? So very not practical at industrial scale. So they're saying, but at commercial productivities, if you were to grow 5 milliliters of that yeast that makes 5 grams per liter of opioids, over several days, you would get 1 dose of pain medication. So 5 milliliters, I don't have a tube with me, but 5 milliliters is like this much this much liquid, right? It's a small amount of liquid, and that's 1 dose of pain medication. So this 5 grams per liter would be reasonable, and it would replace 0.2 square meters of poppy field over the course of a year. So instead of using that 0.2 square meters of land over an entire year to grow poppy, you could instead grow 5 milliliters of yeast in a few days. So that's what they're planning eventually to do, or saying potentially could be done. And so this would decrease the land area required for production by 500-fold. So 500 times less land to produce these opioids in yeast rather than in poppy. This next paragraph is also really interesting. They're talking about the concerns over homebrew opiates, which all of you crazy people in my comment section are talking about with psilocybin and cannabinoids. Of course, I've egged that on. But with opioids, this is really not possible. With psilocybin, it's definitely possible, could be done today. You could produce large enough amounts for it to be reasonable. With cannabinoids, probably not. With opioids, probably not. And so they're saying, since they're producing such small amounts of opioids, you shouldn't be worried about this. It's not economically competitive with poppy farming industrially for either licit or illicit markets. So nobody's going to be brewing opioid yeast at home. Their titers they're getting are less than a microgram per liter for hydrocodone, and Vicodin, the name brand of hydrocodone, you need 5 milligrams of it. So you would need thousands of liters of fermentation broth, which no home brewer would reasonably pursue, right? It would be more efficient for a drug dealer to grow an entire field of poppy than to somehow acquire this yeast and grow thousands of liters of it in their garage or something. And they're also saying that the work here does not provide a recipe for producing opioids in yeast. Because of that, it doesn't directly undermine public health or safety or security, rather. This is probably addressing some concerns with the US DEA agents. By the way, thanks for joining me on the US DEA watch list. Great to have some company. All right, so a strain that converts sugar to morphine would require a 7 million-fold improvement being able to produce these compounds in yeast, you don't necessarily need to produce from sugar or de novo in order for this to be a valuable tool, right? Potentially, there are some enzymatic steps that are more efficient than the existing chemical steps. And so having a way to biologically modify these opioids may be valuable. And, again, there's this $17 million funded company, or they recently got $17 million to probably make opioids in some way. So, of course, there is some value here still. And additionally, there's potentially some chemical steps that have never been discovered or are very economically unfeasible that we can much more easily do with enzymes. And this has been seen before in the biotech industry. It's stupid to completely align yourself with genetic engineering and biology because there's a lot of steps that chemistry is more efficient at, and same thing, vice versa, with chemistry. It's stupid to align yourself completely with chemical production because there's a lot of biological steps that are more efficient than the chemical steps. And so you really need this holistic approach to produce whatever compound you want. Maybe you have to do plant metabolite extraction for the first step, and then you do some biological conversion with yeast or bacteria, and then you do a chemical conversion to get your final product, right? So you need to have this holistic approach in order to make an economically viable strategy to produce your compound. That's all I have for you today. If you liked the video, be sure to subscribe to the channel. Go watch the psilocybin and cannabinoid production in yeast videos if you haven't already. Thank you so much for watching. Bye.